So hi, everyone, and thank you, Carolina, Noam, and Amy, for the kind invitation to share some of our ongoing work with you. So the immune system undergoes continuous remodeling from infancy through aging, and some recent literature has started addressing the question, how do immune cell composition and state differ between children and adults at steady state? What remains even less unknown, though, is how does age impact the immune response to specific pathogens? We've seen some recent work about this in the context of COVID, which is, of course, a disease where adults manifest with more severe complications. But what about dengue and some other viruses? So why dengue? First, it won the poll, as far as I saw before break. But this is a global health threat impacting 400 million people worldwide. Majority of infected individuals remain asymptomatic. Those that develop symptoms have an acute febrile illness, but approximately 5% of those will progress within several days of symptom onset to a severe form of the disease, manifesting with shock, bleeding, organ failure, and sometimes death. In an attempt to identify such severe dengue progressors, the WHO has implemented this set of criteria called warning signs. I'm showing you those in this rectangle. But these are neither specific nor sensitive. So we currently have no clinically approved biomarker to predict severity. Children appear to harbor the greater burden of disease, as well as increased mortality, with greater than 80% of severe dengue-related deaths occurring in children in some cohorts. And they've also shown increased hospitalization with former vaccine versions. The best characterized risk factors for severe dengue progressions has been antibody-dependent enhancement. Viral genetics have also been shown to play a role. But until recently, there was overall limited understanding of other determinants. And the question why children develop more severe disease than adults remained unanswered. So our goal is to try to understand immunological mechanisms that differentially govern severe dengue progression across age groups, with the long-term goal of aiding the development of prognostic tools and perhaps personalized or universal antiviral vaccine and antiviral strategies. So our cohort in Colombia has enrolled over 600 patients, 2/3 of whom are children. And we try to capture patients at an early stage of disease and have demonstrated a progression rate of about 5% in this cohort. A number of years ago, we developed this platform that we called virus-inclusive RNA-seq. This was in collaboration with the Quake Lab, where we were able to capture RNA of dengue, which is a non-polyadenylated virus, concurrently with host transcriptome at single cell level. And recently, we furthered this platform to enrich for rare cell population and incorporated it with 10x pipeline for greater throughput. Combining this with a number of other approaches, we've recently published this, and I'm not going to speak much about our systems immunology model of early severe dengue progression in children. Many of the hallmarks we've identified, which were beyond ADE, involved monocytes. So just briefly, since the rest of the talk is going to be focused on monocytes, I'm going to say that we saw this expansion of classical monocytes in severe dengue progressors, and these cells exhibited some interesting signatures in terms of inflammation, Fc gamma receptors, but yet interferon and antigen presentation dysregulation. So the questions we're trying to answer with the current studies are these hallmarks of severity specific to children? What are the functional impacts? And what are the hallmarks required to adequately control dengue? So here's the design of the current study. We studied both children and adults, and you could see the age distribution. We studied 3 categories of individuals: healthy, controls, is homeostasis state, and here we also incorporated some existing transcriptomic data sets. Uncomplicated dengue represented the adequate responders, and severe dengue progressors, the inadequate response. Once again, we used our VISC-RNA-seq platform and integrated it with serum secretomics, functional assays, and more. I would like to emphasize that this is a collaborative work. Veronica Duran, who's a joint Biohub and Stanford fellow, has been leading this effort, working with a number of individuals, including Desiree Leonard, Bushra Nirong, and Fabio Zanini has been a long-term collaborator from Sydney. And this is all unpublished work. So we normalized each data set and then integrated them into a large data set using Harmony. And what I'm showing here is just the UMAPs and the projection of cell types, 9 of them in both children and adults. And on the bottom, you could see UMAPs that are color-coded for disease severity, dengue exposure, and age. So I'm going to jump straight to differential gene expression patterns. We did observe multiple changes in cell type abundance, and I'm happy to discuss those over a break. So here, to capture the complexity of the data, I'm showing you the log-to fold change between severe dengue progressors, children on the x-axis, and adults on the y-axis. So as you can see, the majority of genes, so each dot here is a gene, show no up or down regulation in either age group. But then if you look at the diagonal, you're going to see concordant findings. If you look at the other diagonal, discordant. And then these vertical and horizontal regions will show us some age-specific responses. Sorry. And here's the same, and here are some of the interesting top differentially expressed genes that we identify in this data set. And I'm going to take you through some of the phenotypes. Again, this is all focused on monocytes. So in this box plot, you could see adults data on the left and pediatric data on the right. So starting with inflammation and chemotaxis, we saw upregulation of multiple genes in both age groups that fall under this category, but overall the upregulation seemed to be greater in severe dengue progressor children than adults. Here are a couple of examples. S100A8 now is data that's broken to individual patients. So S100A8 is a alarmine, which is pro-inflammatory, and you could see significant upregulation in children and not in adults. Similar results with the tetraspanin CD63, which is a migration adhesion molecule. When we analyzed cytokines from the serum of the same patients using Luminex, we saw some interesting patterns. I'm just going to jump and show you a couple of examples here. So TNF-alpha and IL-18, as well as CCL2. These are pro-inflammatory cytokines and chemokines. Were increased at the serum of these patients. So this is now at the protein level, particularly in children who progressed to severe dengue relative to children who had uncomplicated dengue. And this was either milder or non-existing in severe dengue progressors. Now, of course, these could be derived from multiple cell types and tissues, but in our cell-cell communication data that I'm not going to share, we saw that in our cohort, these largely derived from monocyte and dendritic cells. So overall, seems to be increased pro-inflammatory phenotype in children more than adults. Another interesting phenotype involved interferon signaling and interferon responses. So continuing the theme we've started hearing about this morning, what we observed was that whereas IRF1 and STAT1 were upregulated in children, multiple ISGs were downregulated in children, and the completely reverse phenotype was observed in adults. So we were very curious about this and looked at what is the cell specificity of these findings. So here I'm adding another component with different shapes representing different cell types. And we did see that this extends to other cell type. And you could see here this upregulation of STAT1 and IRF in children, whereas overexpression of ISGs, particularly in adults. So we then ask what's going on in adequate controllers. So when you take our uncomplicated dengue and compare them to healthy controls, and here I'm showing you the data as radar plots. These are log-2 fold changes between dengue and healthy. The adult data is in pink, pediatric in orange. And this circle here shows the log-2 fold change of 0. So any data points outside this circle will represent upregulation and inside downregulation. And what you could see is upregulation of multiple ISGs in these adequate responders in both adults and children. And here are some examples capturing patient-specific data. You could see upregulation, again, in both age groups of IFI6 and MX1, which are, of course, antiviral ISGs. What about the signaling? So here we saw upregulation of IRF7 in both age groups, which is an interferon signaling specific marker, and then more prominent upregulation of other signaling factors, including STAT1 and JAK2 in the adult population that responded adequately. And here you could see IRF7 at the patient level with upregulation in both adults and children adequate responders, whereas IRF9 being upregulated more in adults than in children. Moving to severe dengue progressors, so here this picture changed quite a bit, and you could see that there was that severe dengue progressor children showed upregulation of IRF1 and STAT1, but no longer interferon-specific signaling genes, whereas severe dengue progressor adults showed broad signaling gene downregulation. And here's STAT1 data showing that it's upregulated in children, but no longer in adults. The ISG data is shown here. Again, this is the severe dengue progressors versus dengue, and you could see that severe dengue children essentially showed blunted ISD induction, whereas severe dengue progressor adults maintained ISG upregulation despite the signaling decline. So this was a very interesting phenotype, and here's one example with ApoBex3A showing that it's downregulated in severe dengue progressor children, but not adults. So one hypothesis we had is that perhaps there are autoantibodies that target type 1 interferon pathway that could account for some of these phenotypes. This was described in the context of COVID-19 recently and was associated with severity. So in collaboration with Erod Bodansky, Joe, and Mark Anderson, we started to look into it, and thus far we've not identified an anti-interferon alpha-2 autoantibody, but this was just in a subset of the cohort, and we're going to be looking more broadly and also pursuing a FIPSIC analysis. Interestingly, when we look at our cytokine panel, so we observed essentially markedly reduced expression of interferon alpha-2 in the serum of severe dengue progressor children, but not severe dengue progressing adults. So this is very interesting and essentially suggests that severe dengue progression in children perhaps may be associated with loss of type 1 interferon responses. We were motivated to determine whether this phenotype is associated with functional differences in interferon regulation, and to do so, Veronica established this STAT1 phosphorylation assay, which is FACS-based assay where she stimulates PBMCs with interferon and then looks at STAT1 phosphorylation at different time points. And what you could see here in the histograms on the left and the cumulative data on the right is that when we studied dengue patients, these were dengue with warning sign adults in pink as opposed to healthy adults in green. We saw a greater magnitude and kinetics of STAT1 phosphorylation in dengue with warning sign adults relative to healthy adults. So that was suggesting that these patients were primed, they were infected with dengue. This suggested this heightened ISG upregulation that we're seeing in face of low signaling is probably regulatory, and those signaling mechanisms do seem to be working effectively. What about comparing adults to children? This is ongoing work. These are just pilot experiments from the last few weeks that I'm sharing with you. But here we saw quite high variability in the response with two children demonstrating greater magnitude and kinetics of STAT1 phosphorylation relative to adult, and one child is essentially representing no response. So more to come on that, but at least the assay seems to be working, and we can now be more ambitious and try to look at it at a single, at a cell type specific manner. Another interesting pattern that we detected was with respect to antigen sensing and uptake. And here, the pattern initially was not as clear, and there was a sort of a mixed picture with some relevant factors being upregulated in kids and some in adults. So when we looked at dengue adequate responders, what we saw was that there was upregulation of more diverse sensing and uptake receptors in adults than in children. What was striking was this remarkable upregulation of Fc gamma receptor 1A, RIG-I, which is DDX58, and TOLARC receptor 4. So based on that, we hypothesized that perhaps there's activation of the NF-kappa B pathway, since these are positive regulators, and indeed this is what we saw. So in adults specifically, we see this upregulation of NF-kappa B, REL-B, and some other factors in the canonical NF-kappa B pathway. So adults, unlike children, appear to be relying on this pathway perhaps for adequate response. Of course, this requires validation at a protein level. What about progressors? So here, what we observed was that severe dengue progression was associated with lack of upregulation of innate immune sensing in both the pediatric and adult population. But the severe dengue progressor children continue to upregulate Fc gamma receptor, perhaps suggesting a mechanism by which they have a more antibody-dependent enhancement. Yet, what we saw in the NF-kappa B pathway was in line with what we saw with the receptors, and here adults essentially lost activation of NF-kappa B, and you can see that, in fact, there was even downregulation. So this suggests that perhaps severe dengue progression in adults is associated with loss of NF-kappa B activation. We also saw some findings related to antigen processing and presentation that were interesting, particularly downregulation of antigen processing, particularly in adults, in universal downregulation of antigen presentation genes in both age groups. And I'm going to skip the rest of the slides so that I'm going to jump to the conclusion so that we could have time for questions. So clearly, adequate control of dengue infection and severe dengue pathogenesis are associated with different immune landscape across age groups. I think the more we look, the more we will find those differences. It appears that severe dengue progression is associated with impaired innate immune sensing and antigen processing and presentation in both age groups. Yet in children, there appears to be this loss of type 1 interferon response, which is very remarkable, increased pro-inflammatory profile, and potentially more ADE, whereas in adults, there's this prominent loss of NF-kappa B activation. So we think that this may have implications for therapeutic and vaccine designs. And with that, I'd like to thank, again, the people who've done the work, and I'd like to give a shout out to Veronica Duran, who's here today and is looking for faculty jobs. If there's anyone that is aware of any search, we would love to hear from you. And thank you for funding and collaborators who've helped with this.